the break. As you get settled, I'm gonna start introducing our Living on Hydrogen session. As many of you know, this is something Nicole and I started during COVID. It was a webinar where we talk about everything around microbes and underground hydrogen storage. And today, for the first time, as part of the summer school, we're having it in person, which is very exciting. And if for the underground hydrogen school we ever give an award for the furthest traveled, Kelsey will win it. Kelsey is a final PhD student at the University of Canterbury, all the way from New Zealand. She is working in an interdisciplinary group investigating hydrogen geostorage in depleted hydrocarbon reservoir. And let's give her a warm welcome, and the floor is yours.
Tēnā koutou katoa, ko Kelsey tōku ingoa, ko mata o te awa, no ihikatea aho. Tēnā koutou, tēnā koutou, tēnā tātou katoa. I am going to give you a story about the unique hydrocarbon microbiome that we have in New Zealand. So for those of us who might be geographically challenged, I'm from way down here. So this is where we're situated. And New Zealand actually has quite a lot of oil and gas around. We have about 7 different main areas of oil and gas. However, the only economically viable area is located in the Taranaki region here, which is in the North Island of New Zealand, just on this coast here. All of these oil and gas fields are situated around Taranaki Maunga, or the Taranaki Mountain, which is this gorgeous mountain that is on all of our tourism images. It's absolutely stunning, and I got to look at that while I was doing all of my sampling, so I had a great time. So, New Zealand, we are trying to move away from our fossil fuels, and as part of this, we are looking at green hydrogen. So I'm part of an interdisciplinary team looking at underground hydrogen storage, and we are 4 groups: geostorage, prospectivity, reservoir dynamics, the relationship with Wai and Nukatu. So in New Zealand, our native communities are really important to us, as is our flora and fauna. So Wai being water, we need to make sure that we're looking after our local communities, their water sources, and the surrounding areas as well. So there's an entire team dedicated just to this, and also the economics behind the storage. I am part of the dynamics team and the biogeochemistry group. So my main focus of my PhD is understanding the impacts of microbial communities of these reservoirs. So firstly, what is there? There have been no studies on the microbial communities of oil and gas in New Zealand. There was one done in the early 2000s, and they said, we have Thermoanorobacter type stuff, and that was it, which as a microbiologist is not good enough. So firstly, defining what is there in our community, and then secondly, what the function of this community is going to look like. And lastly, how these microbial communities might interact with stored hydrogen. I will only be talking about this today. However, I have some other experiments that I've just finished conducting, so if you want to know about the other side of things, I can also talk about that as well. So for my sampling, I collected 20 litres of reservoir fluid. This came from a variety of sources of wellheads and separators. It was what the companies could give me at the time. I pushed this water through a 0.2 micron filter. That filter is going to capture the microbial communities on the outside of it. I can then take it back to the lab, cut it up, cut all the DNA up, send it away. People do fancy stuff and sequence it. I look at the results. These are some of the areas that I sampled from. So this was one of the separators, this is one of the wellheads, and my samples looked very different. Some of them were really clear like this from the separator, others were full of oil, and then other ones down here came from like a sand capture separator, so it was full of this really fine white sand. I was able to get 3 oil wells and 4 gas well samples. The companies are anonymous and the locations are anonymous, with a variety of different depths and temperatures. For the gas wells, the lower depth well here was sitting around 33 degrees. The rest of them were between 4,500, 4,700 meters, and they were all 65 degrees plus. The oil wells here, the companies didn't have downhole probes, so I could only measure the temperature above ground, sitting around 20 to 35 degrees. However, with later communication with them, they've stated that other wells further away, but of similar depths, are around 50, 55 degrees. So the temperature could be, I would assume, to be a lot higher than that. And the pH was quite stable across all of my different wells that I sampled. So I was able to do 2 different sampling trips. And I have 2 different types of data here. So all of my identifications here are based on whether they were oil and gas and what well they came from, and they're just going to be numbered for the rest of this presentation. So for the Amplicon data, that was oil 1, 2, and actually oil 3, and also gas 1, 2, 3, and 4. And for metagenomes, I was only able to get enough data from oil 1, 2, 4, and gas 1, which was done at a separate sampling trip a year later. So for the microbiologist, you've got like 10 seconds to find your favourite microorganism, and then this is all going to go away. So for the first sampling trip that I did, I got the Section S community, and this gives us an idea of what kind of microorganisms are in these communities. It doesn't tell us anything about them. It's kind of like going to a party and finding out someone's name. That's all you know about them. So here we have gas on the left and oil on the right, and this is a relative abundance plot of the top 20 taxa, or the top 20 genera of these groups, nested by phyla. So the broad colour range is the phyla, which is one of the biggest taxonomic classes that we can do, and then the shade inside of that colour is one of the lowest taxonomic groups that we look at. So we've heard a little bit about microbial communities and what's going to be there. We're anticipating there to be hydrogen oxidizing organisms. We're expecting there to be thermophiles and anaerobes. I'm largely interested in methanogens, sulfate reducers, and acetogens. So we have methanogens. We only have methanogens in oil 1 and oil 2, which was quite interesting. Oil 3 was a separator, and that can have up to 11 different wells being put into that separator at one time. Oil 1 and oil 2 are distinct wells that are only ever sampled from the wellhead. So, only found methanogens in these wells here. We found sulfate reducers pretty much everywhere at very different abundances. And we also found acetogens literally everywhere. So that's great. We have hydrogen oxidizing communities here. We know a little bit about them. So we can take this data and we can do some other things with it. So I can do some metagenomics. For this particular data here, I am looking at one specific gene, and that is kind of like a labeling gene. For the metagenomic side of things, I take all of the genes in that community and I can look at them as a whole. So I can make some really fun trees, some phylogenetic trees, and I can look at the gene content of these particular organisms. So I found 58 genomes, essentially, across the 5 sites that I sampled, and I annotated them with GTDB, which is just a way that you can classify these microorganisms. And we found these ones up here had at least 75% of the genes and the pathway required for methanogenesis. These ones down here had most of the genes or all of the genes required for dissimilatory sulfate reduction, which is going to produce that hydrogen sulfide. And these ones here all had the capacity for acetogenesis, essentially. And finally, when it comes to these metabolic processes, these microorganisms also need hydrogenases, which is one of the genes that they can have that will help cleave that hydrogen. And we found them everywhere in our samples. So a lot of these organisms have the capability of cleaving hydrogen and utilising it for their own metabolic purposes. I was also able to make a heat map, and here we have our samples down the bottom, and we have the different metabolic pathways that we're interested in these different sections here. So up the top we have just general carbon, we have methanogenesis here, we have nitrogen usage, and then also sulfur usage. So what we can see is that, again, only in our oil samples do we have really high abundances of genes that are capable of methanogenesis, or organisms that are capable of methanogenesis. Again, with a really high amount of hydrogenases, which makes sense. They need those hydrogenases for that methanogenesis process. But interestingly, we don't have a lot of high relative abundance for our dissimilatory sulfate reduction, despite seeing them in the initial the name tag part of the study. Unfortunately, these samples can be really difficult to get DNA out of, so I wasn't able to get some of the other samples. So I only have part of the picture here at the moment. So that was that side of the equation done, which was great. However, New Zealand is really far away, and we already know that we have bacteria in New Zealand that are endemic. They are not found anywhere else in the world. So I wanted to see if I had anything really cool. So I did a meta-analysis. I looked at a whole bunch of different international studies. My meta-analysis was quite strict. I wanted them to use the same primers as I did. So when you do this kind of sequencing, you have to choose the appropriate primers for the 16S part of the study. And the primers can tell you different things. So I made sure that the primers of those studies were exactly the same as mine, so they were comparable. They used the same sequencing platform. They were always reservoir water samples. I didn't want contaminated oil or anything like that. Publicly available, and this was all done by keyword searches in Google Scholar. So I was able to find a whole bunch of different studies from different continents for oil, gas, and shale gas samples. And this was my result. So this is a PCOA plot. And the best way to think about it is each of these individual dots is one sample. And that dot has the entire microbial community blended into that exact dot. And the closer the dots are to each other, the more similar those microbial communities are. So these ones here are going to be from the same well, these ones here will be from the same well, and so on. However, you can see very clearly that New Zealand up here has a very, very different community to the rest of the world. This particular type of analysis is an unweighted UniFrac analysis. So UniFrac looks at the phylogenetic relationship between these communities and the different samples. And unweighted means it's looking at the presence and abundance of these organisms. So there is a whole bunch of microorganisms that are present in the New Zealand samples that are not present elsewhere. And this was also statistically validated by a lovely pairwise Pimenova test. So every single comparison between the countries and New Zealand were all significantly different, which was really fun. This leads to further questions as to why they could be different and whether that's important or not. When I was looking into the data, the main difference is that in our New Zealand samples, there is a whole bunch of a type of organism called Comminicella, which can do a whole bunch of different things. It can do acetogenesis. It's a heterotroph. It loves all types of different food, microbial food. And also, we had a really high abundance and presence of Thermoanorobacter compared to the rest of the studies that I looked at. So that was a really interesting result. Which leads me to my conclusions. So what organisms are found in New Zealand reservoirs? We have the classics. We have the thermophiles. We have the anaerobes, methanogens, sulfate reducers. So the general look of our reservoirs is very similar to what we expect internationally. Could they interact with stored hydrogen? Yes. From the metagenomics, from that lovely heat map that I showed you, the genes are there for these microorganisms to interact with the hydrogen. It's not as simple as that. Sometimes groups like methanogens, they really enjoy to use other sources rather than hydrogen first, because hydrogen can be difficult for them to use. They would rather use acetate and a whole bunch of other things first. But theoretically, they do have the genes capable for utilising this hydrogen. However, they are very different to the rest of the world, which means that when it comes to looking at these reservoirs for hydrogen storage, we need to make sure in New Zealand that we are taking a local approach. We are looking at our wells individually rather than relying on a collective data from international examples, because our reservoirs are very, very different compared to the rest of the world. And the implications for storage. We have the organisms there that are going to be utilising that hydrogen. However, they are really, really varied based on where they are. So there were particular wells that only had methanogens, there were particular wells that only had sulfate reducers. So we're able to luckily pick and choose as to which reservoir you might like to put hydrogen into. You might want to put your hydrogen into a well with a lot of methanogens because, hey, they're making a cushion gas. You know, that's better than them making sticky, gross, smelly hydrogen sulfide. There needs to be more research done in this particular area to look at the true significance on these organisms for stored hydrogen, but at the moment that is a start because we were kind of starting from scratch. And I would just like to acknowledge my funding for getting me from the other side of the world. Thank you.
Thank you so much, Kelsey, also for incredibly being on time. So we have lots of time for questions. If there are some burning questions on the interesting New Zealand communities.
Thanks, Kelsey. Super interesting. So you said you have also your enrichment tests?
Yes.
So now I'm, of course, very curious. So are they also consuming in your lab?
Yes, they're making fun stuff. So I was able to get some water that I know had methanogens and some water that I know had sulfate reducers. I put a bunch of it into these 1 litre anaerobic bottles, flipped them upside down, had them at different temperatures, and away they went. I found that the water that I anticipated would have sulfate reduction was making a lovely iron sulfide that got everywhere, and when I took the lids off these bottles, they stunk of hydrogen sulfide. I am still in the process of analysing the GC data. I was able to conduct this experiment last year. However, the company that I sent my sequencing data to didn't conduct the sequencing properly, and I lost all my data. So I had to restart it, which is the restart. However, with that particular experiment, in this exact setting here, I had a change of relative abundance of Ilidosophora vibrio from 10% up to 80%. So I had a massive increase in that particular organism that will reduce sulfur and produce hydrogen sulfide. So I had no methanogens. Well, I had methanogens, but I didn't have any methane produced, which was very interesting. And I tried different temperatures and I tried different parameters. So despite them being there, that might be a really good part of the story, that they are there, but they're not interacting with the hydrogen.
Did you feed them also CO2?
Mm-hmm.
Yeah, you did.
I had a 80-20 blend of hydrogen CO2. So they had everything they needed. They had tungsten, they had everything.
Okay, very interesting.
Thanks.
Any other questions from the audience?
Hi, thank you very much for your presentation. I'm Guillermo from the University of Naples. Just a very quick and specific technical question. When you predicted the hydrogenases, how do you do that?
The what, sorry?
The hydrogenases, the enzymes.
Hydrogenases?
Yeah.
So for that particular diagram, I was just looking for the presence of hydrogenase genes, which was part of that.
Did you do like a blast search or HMM search or?
I used HyDB.
Okay, yeah.
Yeah, yeah.
Thank you very much.
So I know nothing about the microbes, so I had to ask. It's also seen in the field tests that the microbes are there, they got the food. Why are they not working? And I mean, scientifically on a paper, you might not be claiming much, but what's the theory here? Why aren't they?
When I did these particular tests here, I found that even in my controls, so one of my controls, I had the reservoir water, which should have had all the organisms. I filtered all of the organisms out, and I shoved a whole bunch of hydrogen in, and the pH dropped by like 2.5, down to almost 5. So it could be that when we are putting in the hydrogen and other gases, we're changing the environment so much that we're limiting their ability to function. I haven't had enough time to look into the communities of those particular experiments yet. But there is a whole bunch of stuff that happens in those reservoirs. There is competition between microorganisms. There is the fluctuation in their food sources. So it's there could be a lot of factors that are contributing to why some might be producing particular gases and others aren't functioning as we would expect them to.
Great. Very nice. Great answer. Very politically correct answer. Next time I tell a different one to Hassan. And then another round of applause for Kelsey.
Thank you. Thanks.
So I will welcome to the stage Petra. Petra holds a PhD in biology. She is the project manager at the company IsoDetect, so many of you have already met her. And in the past decade, she has led many commercial and research projects on microbial processes in underground hydrogen storage. And today she will talk about one of the projects.
Thanks a lot for the nice introduction, and it's a pleasure to be here and to present you the HYDRA project, dealing with the microbial impact on underground hydrogen storage in European porous rock reservoirs. So during underground hydrogen storage, there are 4 microbial processes known which can lead to microbial hydrogen consumption. So the hydrogen consumption can be coupled to the reduction of sulfate and sulfur, leading to the formation of hydrogen sulfide. Then there is a process known as methanogenesis. So hydrogen and carbon dioxide is converted to methane. And we have the homoacetogenesis. So the hydrogen and carbon dioxide is converted to acetate. So the microbial processes can lead to a loss of the stored hydrogen, and we can also have an influence on the gas quality, as we already listened. We can have the risk of formation of hydrogen sulfide, which is the most unwanted processes due to corrosion effects and souring of our reservoirs. So this is the motivation of the HYDRA project. The aim is to investigate the microbial hydrogen consuming activities and also the interactions with the storage formation. And based on the results, we want to guide principle to mitigate that risk. The HYDRA project started last year and will run until February of 2028. We have 12 working packages in the HYDRA project. So we started with a method alignment, how to sample the reservoir fluids, how we perform laboratory experiments for geochemical reactions, for microbial activities and biogeochemical reactions. And in the last months, we were really busy to sample both fluid and rock materials from porous rock reservoirs in Europe. And the samples are investigated laboratory experiments, so we identify the microbes which are living in the reservoirs in order to get an idea which microbes do we have, what is the potential, what can they do there. We perform geochemical reactions, so what happens if we store hydrogen, are there interactions with the rock material. We have a working package simulating an underground hydrogen storage and investigating how the microbes are stimulated by underground hydrogen storage. And we bring the geochemical and microbial processes together. So how does the rock material influence our microbial activity, and also the other way around, how our microbial activity influence maybe also our rock material. Also results are going towards modeling and also upscaling for the whole reservoir. And based then on statistical analysis, we want to identify the key drivers of microbial risk and how we can deal with the risk. We are in total 9 partners involved in the Hydra project. So the project is coordinated by Martin Fernö from the University in Bergen and Mary Naffiot. We have from Earth Science in New Zealand a geological expert involved. We have, as Isotech company, we are focusing on the microbial aspects of the project. We have the University of Naples involved, also as microbial expert. We have Catriona from the University of Edinburgh, which is also here as our geological expert. We have the Federal Institute of Georesource from Germany for biological investigations, the Karlsruhe Institute KIT from Germany for microfluidic experiments and geochemical investigations. And for the modeling, we have the CSIC from Spain and the Technical University of Clausthal. So far, we collected 21 porous reservoir fluids from 5 different European countries. We have 3 samples from aquifer reservoirs and 18 samples from porous rock reservoir samples. And we have received both, so downhole samples and also samples which were selected at the surface at the separator of the reservoirs. For the rock materials, so far we have 38 rock samples, including both cuttings and core material. And so the rock materials, this is not fresh drilled, but it's from archives. And we have 33 samples from sandstone reservoirs and 5 samples from carbonate reservoirs. Let's have a look to the abiotic geochemical reactions. So this one was studied in high-pressure reactor systems in order to simulate the in-situ conditions, and there are 3 setups. So we have a setup where it's a rock material incubated under hydrogen atmosphere. Then a setup where the rock material is incubated under hydrogen atmosphere using an artificial saline brine. And as a control experiment, the core material plus brine and incubated under helium atmosphere. So what we could observe is that for the private bearing materials, we see some reactions with the hydrogen. So you can see here on the scanning electron microscopy, so hydrogen leads to an alteration of the private material, and we see also an increase of iron in the fluid. This is not the case for the hematite bearing samples, so we couldn't see any change in the porosity and also not in the permeability, and also from the scanning electron microscopy, there is no reaction with the hydrogen. Let's come to the microbial investigation. I selected here 3 samples from porous rock reservoirs. So these are the fluid, freshly taken fluid samples. And of each reservoir, always 2 filters were prepared directly at the field site following the high life protocol. And the fluids are directly fixed at the field site so that we don't have any change of the microbial community, and for identification of the microbes, 16S Illumina sequencing was applied. So what you can see is that we have always a good reproducibility of the two filters, and which microbes will be found. So in blue, we have the methanogenic microorganisms, which are known to convert hydrogen to methane. In yellow and orange, we have microbes which are capable to consume hydrogen and sulfate to hydrogen sulfide. And in red, we have Acetobacterium microbe, which is known to convert hydrogen to acetate. So we could find in several samples hydrogen-consuming microorganisms. Now it's the question, is they become also active if they're coming in contact with the hydrogen? For this, we are running laboratory experiments to simulate the underground hydrogen storage. So we have this small microcosms, and the reservoir fleets are incubated under an atmosphere of 100% hydrogen. At this field site, which I'm showing here, we have a temperature of 45 degrees. And the experiments were run under low pressure conditions. To trace the microbial hydrogen consumption, we regularly determine the pH value, the gas composition, and as microbial hydrogen consuming processes can be really slow, we also applied partially the compound specific stable isotope analysis. The background is microorganisms are isotope selective, which leads to a change in the stable isotope ratio of the hydrogen. And according to our experiments, this analysis is more sensitive compared to the gas composition analysis. In addition, we also analyze the production of acetate and sulfide. So as you can see here in control experiments where the microbial activity was inhibited, the hydrogen consumption keeps stable, and in the setups with the living microbes, the microbes immediately started to consume the hydrogen if they come in contact with hydrogen. And you can also see it for the hydrogen isotope analysis. We see a strong shift in the isotope ratio of hydrogen, which is due to the microbial hydrogen consumption. And what we could also observe that the hydrogen consumption is coupled to the formation of methane. The next experiments are now to bringing the rock material, the brine, and the microbes together, so to study the biogeochemical reactions. And the key questions are the bottle incubations experiment truly representative of our in situ conditions? What role do the rock microbe interactions play in controlling microbial metabolic activity? And if we don't see a microbial risk in the laboratories, does it mean that we have also no risk at the reservoir? We integrate 3 different methodologies to better simulate the real conditions. So we run microfluidic experiments to study these complex interactions between the rock, fluid, and microbes and hydrogen. Then to upscale it, we will run core scale tests on a larger scale. And at the University of Bergen, there is also this metascale fluid possibility to run these experiments. I shortly will show you the results of the microfluidic experiment, which were run at the University of Bergen. So the experiment showed that hydrogen flow can alter biofilm dynamics, and that mineral dissolution can occur locally. and microbes, where microbes and minerals and hydrogen meet. So these are the first results which were run with a pore strain, but we will also run it then with reservoir fluid samples, so have a complex community. With this, I want to summarize the results. So far, we collected 38 rock samples and 21 fluid samples from porous reservoirs to perform these biogeochemical experiments. From the geochemical experiments, pyrite is the most sensitive mineral, which reacted with hydrogen, but not the hematite-bearing materials. So far, we have observed microbial hydrogen consumption in 5 out of 21 reservoir fluid samples. We have in 3 more samples indication for the microbial hydrogen consumption. We have to verify it. And no microbial hydrogen consumption in the laboratory doesn't mean that you don't have microbial hydrogen consumption at the field site. Nicole will show later some results of the High Life project pointing to this. So for the next months, we will focus on the biogeochemical effects on reservoir fluids. And this is then the task for the next year is the risk identification, so which are the key driver for the microbial risk, and to perform the management guidelines. With this, I would like to thank for the EU for funding of the project. I want to thank all the storage operators for the great contribution to this project. And if you are a storage operator and find the project interesting and think, okay, I would also like to be partner of the Hydra consortium, so feel free to contact me. And I thank you for your attention.
Thank you so much, Petra. Now the floor is open for some questions.
Yes, thanks for the presentation. I have a question regarding all your different analysis you do. Do you run them all a single analysis, or are you able to do some sort of a mass balance at the end, which chemical reactions you see in the rock phase, which go into the brine and into the gas phase, and which is consumed? Are you able at the end to bring all together to make a mass balance?
Yes, that's the aim, that we bring it all together. So at the moment we have the geochemical reactions and the microbial reactions separated, but now we're also starting to make it more complex to bring it together, and of course also to bringing then all the mass balance together, which is going to the modeling process.
And this adds up nicely?
Yes.
Okay, maybe I have like a quick question for the on the rock part, because I think it's also a lot of people are quite interested about it. For the rock fragments, eventually you want to go into this mega scale. So how would that look? Like how would this experiment?
Oh, these are the core experiments.
Yeah, exactly,
Yes.
are you using like a rock material that is mimicking one of the reservoirs, or how would it?
So partially we have the rock materials, the core materials, and this can be then flushed with the brine or artificial brine and get in contact with the hydrogen.
Okay. Okay. I think everybody is very relaxed, and but Petra is going to be around, so you can ask her a lot of questions also during dinner time, which I think would be quite nice. Okay, then one more thing. You should cannot get away without your speaker gift. Thank you so much. Okay. So our next speaker is already here, Lydia Krasper. She is a microbiologist at MicoPro with a background in anaerobic metabolic pathways. Her work focuses in microbiology in underground gas storage systems, and she contributes to projects addressing bioremediation and microbial processes in energy systems.
Thank you for your introduction. Yes, I want to give you some overview about the results of the Henry project. Our aim was to investigate the impact of different hydrogen concentrations on microbiological community in 3 Vienna basin reservoirs. The Henry project is the first Slovakia project that investigate the ability of hydrogen storage in depleted reservoirs, and was initiated and organized by NAFTA. And in the first research and develop phase, that consortium is shown here. 9 different partners participate in that consortium, and the main aims of or tasks of that consortium in the first phase of the Henry project are dealing with screening of the potential sites, also geomaterial testing, geochemical testing, microbiological testing, metallic material testing, and in the end also modeling to go to the second phase, the pilot. The Henry project starts in 2022 and run until the beginning of this year, and it's planned to go now in the second phase. MicroPro investigate in that project the microbiological aspects, and therefore I want to give you only a very short overview about our small company. We are only a very small laboratory since nearly 30 years, and are specialized to laboratory services for microbiological aspects, for geomicrobiological, reservoir microbiological, but also biotechnological topics. And therefore, we are involved in some hydrogen storage projects. But let us come to the motivation of studying the microbiology activity in the Slovakia depleted reservoirs. In the beginning of the project, nearly nothing was known about the microbiological community in this depleted reservoirs. And as you already heard in the presentations before, microorganisms are nearly everywhere, and hydrogen is a very good energy source, like for microorganisms like candy. And yes, and that's the reason why we should analyze also the microorganism in each reservoir that are planned to be a hydrogen storage reservoir. And we, at first, we want to identify the microbiological population. Before we want to make some stimulation tests with different hydrogen concentrations. And therefore we need active living microbes, and that's the reason why we make 2 different different methods. At first, very classical, we cultivate the original material from the reservoirs with cultivation broods, and observe the cell growth via microscopy and gas chromatography. And we observed there that in all three analyzed different reservoir samples, we have active hydrogenotrophic microorganisms. They can utilize the hydrogen. And that three groups, also mentioned before from the other speakers, are the fat reducers, methanogens, and acetogens. To prove it with other methods, and we also perform that cultivation-independent methods with DNA extraction, PCR methods, and only amplicon sequencing. And that results confirm our findings with the classical methods. We also find the fat-reducing prokaryotes, methanogens, and acetogens in all three reservoirs, but I don't show that here. We have in all three reservoirs diverse communities. So one of them are more methanogens than the others, and so on. But let us come to our stimulation tests. We done that because the organizers want to know what happens when we have an increasing amount of hydrogen during the conversion to a hydrogen storage site. So, as already mentioned yesterday, in some situations in the pilot, you have only 5% or 50% of hydrogen in your reservoir. And the question is if that small amount of hydrogen has the same impact of the microorganism as when you have pure hydrogen in your reservoir. And that we test with the original formation water samples and original core materials, and at different conditions for easy handling at ambient pressure tests with that small microcosmos experiments, and for some special conditions in that more in situ conditions under high pressure. And what we observed, only some results selected from the experiments here, shown from the ambient pressure test. We observed in samples from reservoir 1 and 2 total gas consumption independently from the amount of hydrogen. At any hydrogen concentration, it seems to be that the microorganism can consume it, and we have a gas consumption. But in the reservoir 3, nearly nothing happens in the time frame. But let us come to in more detail. Here you see, for an example, only for 100% hydrogen, that the gas composition changed during the experiment. As we think that the hydrogen decreased because it was consumed by the microorganism, and you see here that in parallel the methane increases for 1 and 2, but nearly nothing happens for the reservoir 3. And what's also interesting to see here in this experiment, we do not add some additional carbon source, so we don't add additional carbon dioxide to the experiments. That small amount here comes from the formation water or the natural core material. And it's very interesting to see that even when you cannot observe any carbon dioxide, you have a further increase in the acetogenic process. So there are some open questions here. When we have that diagrams, we can calculate on the basis of the hydrogen consumption and methane formation the rates for the modeling, and then we can compare all experiments in dependency on the concentration of hydrogen. And here you can see it for ambient and high pressure tests for all three reservoirs. And the result was that in reservoir 1 and 2, with increasing hydrogen, we have also a slight increasing of hydrogen consumption. And that the average consumption rate at high pressure tests for reservoir 1 and 2 is in the same range as for the ambient pressure test. And it's also very interesting, and I think typical for microbiological tests. For reservoir 3, we have surprisingly a hydrogen consumption and I don't show this methanogenesis formation, but only on a high pressure. So, yeah. So that's the results. I have no explanation for that. We have also a look at the formation water to show if the hydrogen has also an impact of the pH on the acetate concentration before we detect acetogenic microbes, and therefore are they also active and produce acetate. And it is, as Nicole also mentioned, a typical shift in reservoir 1 and 2 was observed for the pH, but not for 3. And we also observed a slight increase or formation of acetate in reservoir 1 and 2, but surprisingly, even when we have acetate in the beginning and in the end of reservoir 3, nothing happens there. There might be, don't consume that. And it's also interesting that we have a formation of hydrogen sulfide in reservoir 1 and 2, but you have to know that all 3 reservoirs have very low amounts of sulfate inside. So that's qualitatively observed, and the microbes are there. But when you have a look at the longer phase of hydrogen storage, probably it don't increase further. Yes. So we can say, based on our results, that we have inside our samples and our reservoirs acetogenic sulfate reducers and methanogenic processes. And to sum up all results in combination of the different hydrogen stimulation effect of that metabolism, we can say that for reservoir 1, lower amounts of hydrogen seems to have an effect on the microorganisms that they cannot consume it. For reservoir 2, also the same, the decrease with a decreasing hydrogen concentration, and for reservoir 3, we have only a methanogenic induction at 100% hydrogen at high pressure. And the carbonate and sulfide pure deposits can limit microbiological activity that we have in our mind for that reservoirs. And with that, I want to give you a short outlook for the results from our batch experiments to field scale modeling from our partner of the DBE. They use our results from one reservoir which was decided for the next pilot, and model with that what happened during 3 cycles of injection and production. And yes, you can see here that the acetate and methanogenesis increase over the 3 cycles of 2 planned or modeled years. And with that, I give you here also a short overview of all the challenges that are with modeling. So the modelers also included the geochemical results and the results of the other groups, the other partners from the project together, and make that model. And in the end, the hydrogen purity improves over successful cycles, but the model said that 3 cycles are insufficient to reach the high recovery of hydrogen. But when you have more cycles, 5 or 10 cycles, then the model said that you have high purity for that reservoir. And with that, I want to come to end and thank you for your attention.
Thank you, Lydia, for the great talk. And we are open for some questions.
Thank you very much for the talk. Markus Bichler from RAK. I have actually 2 questions. One connected to slide 7, because I saw how these water samples look like, and I would just be curious if you took the water samples at the top, in the middle, or the bottom of the column. Because there was a lot of fine material in there, so I guess you took them at the bottom. Yeah, okay.
Yep.
And the second question would be actually a comment. I mean, I'm not a microbiologist, but I am fortunate enough to listen to a lot of microbiologists. So my comment actually goes towards reservoir 3, because microbiologists always tell me about acetate and how acetate can be then converted by whatever microbe, I don't know, to methane. And now seeing the whole thing with you saying that there is a higher pressure of hydrogen necessary in order to get the reservoir 3 going.
3? What? Wait, which slide?
Last one. No.
Last one.
Yeah, relative. I'm already done with last. Yeah, let's take that one, but it doesn't matter.
Okay.
You showed that for reservoir 3, with a higher partial pressure of hydrogen, you then finally get the consumption and you get the methane generation. So could that just be a question of partial pressure? Because I think if there are like massive amounts of acetate in there, which you showed that the biggest amount of acetate was in reservoir 3. So they might just need the kickstart, the microbes, to have enough hydrogen available so they could convert this acetate into methane, which you also showed. In higher partial pressures, you showed a lot of methane in this reservoir 3. So maybe it was just the partial pressure of hydrogen that was too low in the other cases in order to get the acetate, however, genesis going.
No, I think not. But the acetate in the high pressure tests don't change. So we measure in the beginning and in the end the acetate concentration, and it's the same level. So
Okay,
maybe
I saw just like a
nothing-
small shift, and I thought, okay,
Maybe
it might
yes.
already be enough. As I say,
this
I'm not a
small shift-
microbiologist. I'm just seeing things and listening to people.
But I think that small shift cannot result in such a high methanogenesis production we observed. But maybe all three different processes take apart and connect to each other. So we only observe the sum of all metabolic processes. So I cannot give you a clear answer because the acetate can also produced and consumed by the methanogenics, and the concentration we measure is the same in the end. You know?
But I said it. Okay. Okay. I thought I saw a shift and thought, okay, maybe do a material balance to check. But if you say it's the same, then it will be the same, I guess. Thank you.
Thanks.
Okay, thank you for your presentation. Franziska Steger from RAG. My question is related to slide 9 of your presentation, so the next slide. So you said you did not add any other carbon source other than the CO2 that was already present in the beginning, but there's still methane formation. So do you have any idea where the carbon could come from here?
Yes, the core also contains some carbonate, so it comes maybe from that. But it's interesting to know that even when it comes from the core, in the end of the experiment, other project partners analyzed also if the rock material composition changed. And that was not the case. So,
So many questions. Oh yeah, one.
Hi, the microbiologist that Marcus listens to, and sometimes maybe he shouldn't, but in the last slide, I saw the cytogenesis in the different H2 percentage incubations in H2 partial pressures. Could you please go there? Because I'm not really sure that I understood the slide correct, so maybe the question is irrelevant, but...
does this one?
The very last one with the...
Oh, the very
overview,
last one.
So, previous one, sorry. Also, one more back?
No, okay.
Oh, no. Well, with the plus minuses. With, yes and
okay,
no.
okay.
So,
Summarization.
do I read correct that the cytogenesis at lower... so, yeah, exactly. At the ambient pressures, at all kinds of hydrogen concentrations, you actually do not observe a cytogenesis, whereas then at high pressure, it appears. Right. Do you have an explanation? Because for me, this is very interesting and new. I've not seen this before.
Not really. Not really. Because our experiments from other experiments are normally that you get more results under low pressure, because then you have one less stress factor for the microbes than at high pressure. That's the normal thing we observe normally. But yeah, in this case, it's I have not answer for you. Sorry.
No worries. I just think it's quite a remarkable outcome.
Many thanks again, Lydia. So now is the perfect time for all the microbiologists to talk to Lydia during coffee break and figure out some of the questions. Thanks. So we have a very short coffee break, so 10 minutes just to stretch out your legs and get some refreshment. So see you back here in 10 minutes. refreshed and more ready for more microbiology. Our next speaker, I think some of you already know him quite well, is Artur Zadurian. He is an environmental microbiologist, studying microbial communities in underground hydrogen storage systems. His work focuses on understanding and managing microbiological risks, and he is actively contributing to the development of safe hydrogen storage infrastructure. And the floor is yours.
Thank you for the introduction. Happy to be here. You might have already devised, probably from my last attempt to sound funny, that most of my microbiological work is related to RAG Austria. We've been supporting, from the microbiological side, development of renewable gas technologies underground. When I was preparing this presentation, I thought I will just sort of collect the last developments that we have, but then realizing that in the context of this also summer school, maybe I should take sort of a step back and maybe provide you with a more general alternative view on microbiology in underground hydrogen storage reservoirs, which is related to the concept of geomethanation, where basically in depleted hydrocarbon reservoirs as well as salt caverns, the resident microbiology is by more or less default seen as a LOSC, because hydrogen can be consumed by these microbes through a variety of processes that have been already many times shown by excellent colleagues today. Homocytogenesis, sulfate reduction resulting in hydrogen sulfide production, also iron reduction, which is probably less relevant in our systems. But sort of the star of the evening today, the methanogenesis, has been sort of the idea behind this presentation is to move from risk to opportunity and look at hydrogen consumption by microbes when provided also, importantly, with external CO2, the conversion to methane, which not only could sort of serve the couple of advantages, which are methane being a grid-compatible molecule and having higher energy density, as we discussed yesterday extensively, and also this needing the molecule of CO2 for this conversion process facilitated by the microbes. We can sort of also couple hard-to-abate industries such as steel mill production with this hydrogen, renewable energy hydrogen production, and turn the underground hydrogen storage, underground gas storage facilities into really a large, huge biomethanation reactor, or geomethanation as we call it, which is depicted here on the right side of the slide. It all, however, started with the first controlled pilot of storing hydrogen underground with RAG, of course, in a depleted hydrocarbon reservoir, Lehen in Austria, which is about 1.6 million cubic meters of volume. And in there, 120 cubic meters of hydrogen were co-stored with natural gas and not with CH4, an important mistake I've made here. The reason why it is important is because natural gas actually naturally contains traces of CO2. And even though this was originally this 285 days co-injection storage and production of hydrogen to show pretty much for the first time that hydrogen can be stored safely, which has been also discussed already many times since then. That in this first trial, about 85% of hydrogen was recovered. And when we calculated all the non-biological routes of hydrogen retention, such as diffusion, dissolution, we came up that up to 6% of this hydrogen must have been converted to CH4. As you know, we cannot really measure the CH4 because it's co-stored in CH4 and the machines are not, also have been discussed earlier, machines do not measure such incrementals. when that was seen, and it sparked a couple of projects as well, and part of one of these projects was long-term conversion of H2 and CO2 in the lab, in these first-generation bioreactors that we have, mesocosms. 32 cycles over 357 days were performed. On the right side is the sort of the main figure, I guess, of the paper, where we can show that the dots are all the cycles, and you can see that on average, so consumption goes to the bottom, production goes to the top, methane is produced, carbon dioxide and hydrogen are consumed. It's not shown here, but the conversion of this 2-liter volumes. Inside there is also a core that comes from the Leyen, from the actual reservoir. So the microbes are actually colonizing cores with a little bit of this brine that was provided. And the conversion happens quickly in these 800 milliliters of gas provided at 40 bar pressure. The conversion takes less than 5 days. It was stable long term. No deterioration of the signal was observed. So we could, and importantly, the methane evolution rate was calculated at this value, which the important part of this value is that it is comparable to actually pure cultures in the lab in much more controlled environments. So this goes to say that the potential of this conversion in the field can reach very high methanation rates, such as those that are commercially used, for example, in anaerobic digestion or other methane-producing technologies that are already implemented today in other fields or in adjacent fields. In this original figure, the idea of coupling CO2 from hard-to-abate technologies was presented, and the effort in this direction was to actually get some CO2 from a Voestalpine steel mill, which is in Linz, Austria. It's an actual steel mill. An amine scrubbed CO2. It's not dirty CO2. It's important. We tried it with dirty CO2. It doesn't work so well. Had to clean it up. And we ran in this, I think it's 3rd generation now. It's a much larger reactor. We have 1 meter. Inside there is a core. It's fully equipped with a core that does not come from the Leyen. It's normal Berea core with about 2 liters of volume, gas volume. 1 meter long, 10 centimeters in diameter. And we ran basically a series of 2 times 6 cycles. For this conversion, the units are a bit different, but the point here is to see that the conversion from, so first 6 cycles were run on synthetic CO2, the second 6 cycles were run on Voestalpine CO2, the CO2 that comes directly from the steel mill plant, and you can see that no deterioration of the process can be seen. There's even a fancy unnecessary statistic fitted in there. So the take-home message here is that we can actually use CO2 from steel mill, from in fact various sources. And we can produce methane from hard-to-abate CO2 that can be, in fact, also, if coupled logistically correctly, can be really used on site, and this sort of opens route to this sustainable carbon cycle involving renewable hydrogen. Over the course of the years, and I guess all the microbiologists and operators will know this very well, the understanding grew that sampling of the reservoir wells is a challenge, because we don't. It may not always be representative, but we also don't know how exactly to make it representative. What exactly is unrepresentative? What kind of different parameters influence this? Usual sampling involves samplings, such, this is the schematic, without Marcus it wouldn't exist, of how specifically the Leyen well works. Here's the sump, here's the actual, I guess, well, and usually sampler goes down here, a baler takes this water up, and this is what happens. The perforation is here, so the reservoir is above the sump level usually. So we used this fact to design a sampler that in fact involves rock cores, which also, by the way, comes from drilling of this particular reservoir. So it's as representative a rock as possible, which then, this is a post-hoc division, but it turned into sort of three fractions. The rock that was not in, the idea is that when the sampler is attached here on a gauge above the reservoir, when the drawing of the reservoir comes, when the production of gas happens, reservoir liquid or at least droplets are produced, which then seed this chamber inside of the sampler. And within this chamber of the sampler, we have now three different fractions, which is rock core that is wet, the chamber water itself, and rock cores that never were particularly wetted. Or at least the wetting was transient. We cannot really tell because we don't have cameras down there. This to test how useful such a device is, we went to look at the microbial community data over the course of geomethanation usage of this Leyen well. And what you can see here is the original sample in January 2020. The original community, so this is a microbial community structure, just like what you've seen before, but we only care for two of these guys, and I will tell you now why. So Methanobacterium, the purple ones, are the classical hydrogenotrophic methanogens, which facilitate the whole geomethanation idea, the whole story of conversion of H2 plus CO2 into methane. This was a very nicely geomethanating community that we've originally observed. And what happened is, the operators especially, I guess, know this, that methanol is used when you produce a gas, when a gas from high pressure to low pressure expands, it cools down very quickly, and that results in hydrates. Is that the correct word? Yes. In gas hydrates, and to prevent this, methanol is typically often used. And methanol is such a volatile substance that I guess no one really, it's almost not feasible to think about what's going to happen to methanol, because methanol is so volatile, it's just going to be produced with the gas. However, for one reason or another, it appeared that the methanol ingress happened during these two samplings, and the orange line here shows methanol measurements behind this graph, which already a couple of months actually later, we can see very high amounts of methanol in the sample, which we initially didn't really notice because we were not looking for it. But then we started noticing this Methanosarcina genus of also methanogens, in fact, who also can use hydrogen and CO2 to produce methane, but in fact they like methyl-based compounds, of which methanol is the simplest. Methanol is a methyl group, pretty much. And so when we saw this, later on only we kind of added things up together. And over the course of time, we can see that actually Methanosarcina quickly sort of outcompeted the Methanobacterium. And that was a bit confusing, and all other things came together. And only very much later, when we put things together, we introduced this biosampler in, November 23 for 5.5 months until end of March 24, and incubated it as a sampler. These continue to be samples. This is all water samples, continuous water sampling. And these are the microbial communities which we got from the rock sampler. If you remember, I mentioned that there are three fractions: rock saturated, which is the wet rock, the rock non-saturated, which is the the rock on the top, and then the chamber water. And what you can see here is that even though the microbial community in the sump looks like this, clearly sort of carrying the legacy signal of methanol contamination, the rock fractions, all three, were essentially devoid of this legacy contamination signal, proving that this is a good use case for such a sampler. And also now, I saw a very similar development from Petra and colleagues, and another use case could be indeed when you don't have a sump at all, when only some droplets, micro droplets, can be produced, or even, I don't know, free cells are produced, and Petra could show that such a sampler that is hung in the well can actually also host the community and be inoculated by the community. So yeah, I guess it's a nice step towards more representative and accurate microbial sampling. And I think that was what I chose to show today. And the summary is, first of all, that technology of geomethanation could be shown in the field. There's a lot to do in terms of how efficient and potent and quick it is in the field, but we could show that in the lab, the conversion is as rapid as any other methanation technologies, biomethanation technologies now available. We could use industrial CO2 as a substrate for such process, and that the rock-based sampling quite clearly shows advantage in particular scenarios such as the one that I just presented. And overall, in total, I think it's safe to say that depleted reservoirs, probably as well as salt caverns, may serve really as large-scale and low-cost bioreactors for renewable methane. No huge infrastructure on the top. Yeah, and with this, I would like to conclude my talk, my sharings today with you. Oh yes, of course, I would like to acknowledge all my co-authors, part of which are here also, the RAG colleagues. RAG is also obviously responsible for all the site and partially the funding. Most of the data I presented but is from this carbon cycle economy demonstration, CCED consortium or project, flagship project of Austrian Research Promotion Agency and Climate Energy Fund of Austria. Thank you very much for your attention, and I'm happy to share with you, answer your question.
Interesting talk. Is there some questions for Artur? And also Emil, the RAG colleagues, I guess, also.
Thank you very much. Very good talk. So you said when you tried the biomethanation that with dirty CO2 it didn't work. Could you elaborate a bit? Did you find out what was the problem? What was inhibiting with the dirty?
No, so I was actually not there yet when that happened. I only analyzed the data of that time. In fact, so there were two setups. One setup was dirty gas but cleaned out of oxygen, which is very important because this is an anaerobic community, meaning these microbes can only really function without oxygen around them. And one was not cleaned at all. So when there was a little bit of oxygen, so everything died as expected. However, a sort of medium clean gas. I mean, it was only really cleaned of oxygen, or it didn't have oxygen in the first place. There are different gases, off gases from steel mill. In that case, in fact, the methanation still did proceed, but at a speed 3 times lower. So, yeah, which is possibly sometimes also a feasible scenario, maybe, I don't know.
Okay. And that's some other questions. So, unfortunately, oh, okay.
Hi, thanks for the presentation. I was wondering, why do you want to change hydrogen to methane?
Well, there are two main reasons. The first reason is that imagine we transition to some sort of even hybrid hydrogen consumption in the sort of first world, or the countries that are leading it: Norway, France, Germany, Austria, I hope as well. How quickly that would happen elsewhere. So the sort of the lagging, so the heterogeneity of development in different parts of the world probably call for some sort of bridging solutions, and this could be one such. Because, yeah, to build hydrogen infrastructure in a country that is slightly behind it would not be possible. We saw this in the game today, right? You need to introduce UHS, you need to put more resources, and maybe not everywhere it's possible for a long time still. And also, importantly, in fact, for such high energy demanding industries like steel mill, the higher energy per unit volume of methane compared to hydrogen is a decisive factor in many cases, I imagine, or that's what we assume.
Doesn't it make sense to just, your points are completely valid, and I understand this. Your points are completely valid, and I understand them. The thing that I don't understand that why do you, if you want a green solution here, why do you change the hydrogen to methane? Why? And because of,
it remains totally green. It depends on the essence of hydrogen, and that's it. So if the hydrogen is green, this technology will be green. If the hydrogen is blue, this technology is blue. Because for one molecule of methane burnt, you produce one molecule of CO2, and to produce a molecule of CO2 in this biomethanation fashion, you also need to consume one molecule of CO2, making it essentially a zero CO2 cycle.
Okay.
If I may, I just support it a little bit.
Thank you.
Because there is a decisive factor, which is, at the end of the day, we have not yet decided whether there will be a hydrogen market or maybe another energy market that we cannot know. But if we're having the possibility, theoretically, to use methane, it's like you could go on tomorrow. There's no changes to the infrastructure at all. But I'm with you that this hinges on prices. That's the question. What does the market, what is the market able and willing to pay? Is it willing to pay more, but then investing less in infrastructure, or is the infrastructure cheaper? That's for the market to decide.
Now that green hydrogen is so expensive,
yeah, it will be even more. Yeah, I'm with you. I'm with you. So using the expensive green hydrogen to generate even more expensive CH4. It's a good question.
Okay, great. Maybe just a quick question from my side. I'm just curious, which pure culture of methanogen did you compare for the rate?
I was anticipating this question, and I do not remember.
Oh,
I wrote this 2 years ago.
Okay.
But I promise you that I did my research.
Yeah, it's okay.
I just don't remember,
Can be
honestly. Most likely, you know what? Most likely it's Methanobacterium. It must have been.
Okay.
Because that's what we use.
It's okay. There's no right answer. Okay, then. Thank you so much for your talk. And another round of applause for Artur. Okay. And our final speaker today is somebody I never met before in my life. And is Nicole. She is a senior researcher at NORS, specialized in geomicrobiology. Her work focuses on microbial processes in underground hydrogen storage. She brings experience from both academia and industry research, and the floor is yours.
Thank you so much. So you're almost done, with the microbiology, and then you can really say now you're halfway towards microbiologist studies, So, and I promise you that is the last microbiology-themed presentation in the summer school. But, and also, I just wanted, to take up this chance to present the results of our HyLife project, which, has been now going on for the last 3 years, and I'm immensely proud of it. So that's also the reason why I took the liberty to put myself on the agenda. It's great when you make the agenda, you can just put yourself on it. So because I want to show you the amazing results. So we already heard about it. I think we're okay now with understanding why microbes are important for hydrogen underground storage, what they can cause and problems and so on. So jumping over this. So when we started HyLife with a consortium of different microbiological labs, we really wanted to understand, okay, we see microbes are a risk, but how big is the risk? And is it always the same? Is it for each storage site a risk or just for a few ones? So we really wanted to go out, unite our strengths in microbiology and then define a standard together. And that all the microbiology labs involved exactly do the same methods, so we can compare the results, go out, do lots of sampling, or I phrase it differently, ask lots of operators to give us samples and then test it in the lab. We also did tests like how can we mitigate microbial risks? How does this mitigation efforts actually influence your business case of a hydrogen storage site? Because we now heard, okay, there could be then H2S cleaning systems needed. What would be the price of the biosite if you want to use it? All this will at the end also affect the costs of your storage. So this is also something we are investigated. And at the end now, we want to start a database on all the results, which will be open to everyone to use so that they can see what kind of microbial communities we find. And then the final deliverable, which we are writing right now, is some kind of guidelines for the operators. What should they look out for? What did we find now as an important part coming really from the sampling to the analysis? And then give you the overview, finally the overview of like the presence and/or, because it's not always connected to activity. So first thing was really finding the methods, because when we started HyLife, there were a few laboratories doing this kind of things. You already heard a bit, and every lab and some project has their own method of sampling, has their own primer system, own system. Like I use the big bottles now, I use the small bottles. So we really wanted to say, okay, no, we sit together. We had lots and lots of online meetings, very long and painful online meetings, because we really were fighting, okay, who has the best method and how can we make it, a good method to use for all, so that all labs has the possibility to do it and that it's still, especially with the sampling, doable by the storage operators. That is really critical, because we can come up, of course, with super fancy sampling methods, which are just, when you then give it to the operator, are not doable, because you need, a degree in microbiology, and that's maybe a bit too far and to demand. But we found that, we made videos. You can find it on YouTube. If you also want to take a sample from your storage site you have in your garden. And we also designed the sampling kits, and we designed the SOP, and then we sent it to operators which were willing to take part in HyLife. You can see just a few examples. And the most important part was always that we wanted to get oxygen-free samples. They then, at the end, looked a bit like this. It was always some kind of liquid in a bottle. And then the first thing we always did, look if the headspace contains oxygen. And it was very critical that we don't have oxygen in that. And then what we did with all this brine, so the brine went to the 4 different labs which were involved. And then we, of course, first checked the chemistry. We looked what the liquid contains, just typical water analysis, all the factors which are important for us, like pH, salinity, and so on and so on. We also did a DNA fingerprint from each site, and there the really important part was that the operator also did that for us. So we had a separate set of instructions just for the DNA filters, because from our experience, if you wait with the sample coming to the lab and then do the DNA, it's already completely different. So you need to really get the DNA sample on site, like within an hour after you got the sample out of your well. That's just the best. And then we also did enrichments with the brine, with different gases, of course, hydrogen, nitrogen, and we always had controls. We always used the field temperature the operators gave us, and also 30 degrees, just, because we wanted to see maybe 30 degrees is still something for them. And we also had a setup where we added nutrients, a very defined set of nutrients. And then we followed the growth. normally around 6 months, but for salt caverns, 1 year or even longer, because we realized with salt caverns, sometimes a year is not enough. Some results are actually after 400 days and longer. And what you then have is, like you see this, you have a set of bottles on their head to avoid loss of hydrogen over the incubation. And then we sampled over time. If you're interested in our high life methods, there's also a publication on that, which describes everything in detail. We did a round-robin test initially between the labs with a defined culture, defined media. Everybody did the same. You could see we didn't get the same results, of course, but that actually helped us to refine the method again, and also it highlights that working with hydrogen is tricky, that's one thing. Working with microbes is tricky, and so the combination is already something where you get different results. So you can look it up online, and we hope that our methods, we worked hard on it, will be also useful for other projects. So we're happy to see that other projects already pick it up, and then it can be developed even further. This is now the final results of all the samples we obtained. Overall, 58 samples from 22 different operators in 13 countries. So we're very, very happy about that. We had, as you can see in the pie chart, 17 salt caverns, and then also some leaching water of the salt caverns, which has a very specific question behind it, which I will not go into detail. We also then have 26 porous rock reservoirs and 11 aquifers. So this was a very great success for us, and a few storage operators are here, so I want to say thank you again for this. Without all these just in-kind provision of samples, this would have not been possible. So now let's look at the summary results we have. So these are now the average site characteristics we found for the different sites. So you can see they were already vastly different when it comes to temperature. So for the gas reservoirs from 20 to 150 degrees, salt caverns were more moderate, 14 to 50, and then the aquifers. Then, of course, with the salinity, the salt caverns are very saline. What a surprise. And then we also had a huge variety in pH. Sulfate, again, the salt caverns come out, which is also known as very, very high sulfate content, and vastly different amounts of organic carbon. So really every site was, different somehow. It depended a lot on what the operators were doing before, what kind of history we had. And we learned also with this, because sometimes we obtained samples and we really were shocked to see what we got. And then always after discussions, we actually sometimes found out that chemicals are regularly, there's a regular input of chemicals into different storage systems, which maybe are completely normal for operators, but not for for us. So you might see that, for example, we have salt caverns at a pH of 11.6. This is, of course, not natural. That would never happen. That is an operator who's on purpose adding NaOH to increase the pH. For certain reasons. So, but of course, this is always, you need to find this out and then always see what that actually means for the microbiology. So here is now our fancy, beautiful graphic. You already now saw bar charts with microbes. I will not go into detail because you can see we have lots and lots of data. This is now also just the average of the replicates. But what I want to show you with this is that we actually found microbes in each single reservoir, each single salt cavern, even the high temperature fields, we had microbial DNA. So sometimes the concentrations were very low. To be honest, it was very challenging. But we made it work. We got a DNA fingerprint for each single site. So we can already say, when people ask, like, do I have microbes in my storage? I can already, yes, you have. At least on DNA, yes. But what does that mean now? At least now we have this beautiful information. What you can do with this kind of DNA information, you already heard a glimpse of it. You can use DNA information for metabolic prediction. That is a bioinformatic tools, available different ones where you can then draw information. Okay, what kind of microbes do I have? What could they potentially do? Within iLife, we have a very clever postdoc at INRIA in France who developed a new pipeline called TABECI. It's also published. You can look it up. You can also use it. It's available online. And then you can input your 16S DNA data, your fingerprint data, and it gives you predictions. Again, I will not go into details. You can see, okay, we have like, do we have fermentation? You know, classical fermentation, they use carbon, and like everybody can do fermentation. That's just normal. But then it gets more exciting, you know, can look at methanogenesis or all the sulfur pathways. And then you see here and there, you know, a sample is popping up and the other staying blank. So this gives us some indications. This is, of course, a very academic way also of looking at it, because this shows, especially acetogenesis, super common. We almost have it in every sample, but it's based on predictions. It's we're not sure if this is really true. All this information is drawn from relatives, and sometimes we actually don't really know what we have. We have sometimes a family name, but what does a family name tell you? You know, I mean, everybody who has a big family, you know, now imagine you're just put into a pot based on your last name. And then, oh yeah, this is like Müller, yeah, good in cooking. You know, that we know that, but we have to do it sometimes because we don't have more information on them. So what we then, of course, wanted to see is, does the activity really occur? And for this, we did cultivation tests. As I said, different temperatures, different setups. And I just now want to show you some examples, how this looks like. You already saw similar graphs that we have over time, hydrogen consumption, and then we see, for example, methane production. This is now a very easy, understandable example. Sometimes it wasn't that straightforward, but for presentation's sake, I think it's just easier to show the nice data. But now all combined, now imagine we had to do this for all the samples. It was a huge effort for all the labs, which I also acknowledge highly. This is now the summary, what the consumption. So from our 46 brine samples, because for some we just got DNA, 46 samples, 17 actively consumed hydrogen in the lab. This amounts to around 37% or 40%. You can also see when it comes to salt caverns, porous rocks, or aquifers. So for each type, we found at least one being active. Especially the porous rock reservoirs were more active or in higher chance to be active. For the activity, we saw that salt caverns only just showed sulfide reduction, and they were extremely slow, painfully slow sometimes with activity after 400 days. Gas reservoirs were dominated by methanogens most of the time, and we always got this very marked pH increase. And again, acetogens, they're just very tricky to assess. Because we really don't know if they're active or not, because acetate is just so transient sometimes. So some cautious advice is, of course, some salt caverns we just incubated 300 days, and then we thought we're done. So now that we, of course, had one appearing after 400 days, we might have missed some salt cavern activity. So we should have probably incubated them longer, but at some point you just also want to be done with some kind of enrichments, but that means we are really at the lower estimation limit for salt caverns. We also know many microbes cannot grow in the lab, they just don't like it, and we also had a problem that some of our samples were exposed to air. it's just sometimes not avoidable. During the sampling was some error in maybe the gassing. It is just how it is. So this could also be leading to some inactivity. So now we are, of course, having lots of chemical data, DNA data, enrichment data. So we're now in the final stretch of doing the statistics and so on. We have our first publication also out where we looked at the first set of samples we had, and we can, of course, then combine, okay, we have chemical clusters, microbial clusters, how do they relate? Can we link it together? So this is now all what we are doing right now. What we already can say is that for these porous reservoirs, which are often carbonate-rich, we see they are dominated by methanogens, syntrophs, SLBs, fermenters all the time, and they're really already very prone to consuming hydrogen. As soon as you have fermentation, you will have hydrogen, and so they're already used to hydrogen, so if you give them more hydrogen, they will be just active. For salt caverns and high saline samples, they were more dominated by more generalists. So the microbes which can really do everything, because I guess they're more stressed there. We never found methanogens, but very often nitrate and sulfide reducers. And we also had some samples which are complete outliers from all the predictions, and that was mostly, again, human activity. Something was different, some chemicals. And of course, when you then have a community and then certain chemicals, your predictions are just not usable. It's just like, you can forget it. So everything we do with our storage site has an effect. So summarizing, what does that mean? We find microbes in all the samples, and we have hydrogen consumption potential in all of them, to say from the DNA, but we found out of 46, 17 to be active. And we, again, I stress it again, the field history, the chemical input, everything you do will have an influence. So that really also means if you transfer, the knowledge from one side, you have to be careful to the other side. Even if it's within the same operation, the same reservoir, even sometimes just the history of one well might be so different that you cannot transfer your findings from one well to the other well. Marcus, you're smiling, you know what I mean, yeah? So we're still now thinking about how do we monitor, how do we mitigate, so this will be something in the future. So the open database is almost done, so all the microbiologists can be happy to have a look at that when it's ready. You will also be able, also the non-microbiologists will be able to do some statistics inside, some diversity and so on. So this will be published very soon. And because HyLife is ending now officially, I also want to invite you to our final conference. This will be in September in Leipzig, where you then have one full day just of all the results of the HyLife project. We obtained lots and lots of more of results, which I cannot squeeze into here. So if you're around in Central Europe during September, then just come by. We would be happy to show you our results. And with that, I want to thank lots and lots of people. Of course, all our funding agencies. It is under the CETP partnership, which is under the EU umbrella. And thank you for your attention.
Great, really nice talk. So we're open for some questions. For example, if you want to know more about the chemicals.
Yeah, no, that's fine. No details.
Hi, Stefan from IFE. You didn't get any samples from offshore? There is no drive to store hydrogen offshore? Like
no, in HyLife we didn't have offshore. It was really focused on onshore fields. There's some interest also in offshore. We have another project, Verena has a poster on it, where we got offshore field samples, but offshore hydrogen storage is even further in the future compared to onshore. Everything gets more complicated, of course.
Hi, thank you, Nicole. Isn't there a motivation to get to know these microbes? Chose the correct word, I assume. The better. They come from quite deep underground. We don't know much about them. From the fundamental science point of view.
Yeah.
Don't we have to, also check if they're able to do other stuff other than?
Yeah, of course. I mean, it's super fascinating. We find microbial groups which are so undefined, especially in the salt caverns. We sometimes can just group them to bacteria. That's all we know, because there's no relative closer than it's bacteria. Of course, it has lots and lots of fascinating potential. What can they do? What are they doing there? How do they get there? How do they survive? Of course, this is a completely different type of project then, which needs completely type of funding resources, which I'm always happy, if somebody feels to have that. So of course, yeah, it would be great to do that also.
Hi, Nicole. Thank you very much. Fascinating for me personally. Actually, something that I probably should have known, but it again struck me as something new. So you said you actually do not find almost any methanogens in salt caverns, and then you have the sulfate reducers. That's explainable. You have a lot of sulfate. Do you also have a lot of nitrate? And then that's why the nitrate reduces. So why would you, basically, you would say that, just you have someone who outcompetes them by delta G, or why do we not find methanogens there?
Yeah, I think, so we only had one salt cavern where we had methanogens present, and that was caused by methanol. So they were not hydrogen neutrophic. They were, yeah, exactly, methanol. So it reoccurs all the time. But actually for the salinity of salt caverns, there is no described hydrogen neutrophic methanogen. So we know none. There's never been one isolated. There's certain environmental hints that it is possible, but until now we have not identified any methanogen growing at this salinity. So, and we also couldn't see anyone in the salt caverns we had.
And so the salt caverns are even saltier than, for example, some the salt lakes, like the classic environmental.
They're just saturated and above. It's just, I mean, it is brine within salt. You know, it's as salty as it can get, and it's extremely nutrient limited. It's dark and anoxic. Very often, these open salt lakes are driven by sunlight. This is, of course, in a salt cavern not possible.
Yes.
So it's also a factor. So I think they're probably there somewhere. We haven't just found them yet, but also we couldn't identify them.
Thank you.
Great. Thank you so much. And another round of applause for Nicole. And with that, we are closing our Living on Hydrogen session. So maybe you'll return again next year, so you don't know, but it'll probably be online again. I would like to later, when we break or when we go away, I would like to invite all the speakers to on stage for a little group photo. So if you can just stick around the speakers, that would be good. And some housekeeping for this evening for the restaurants. As you already got from the email, the restaurant will be on top of Floyen. So you also have the QR code to go up to Floyen. And the funicular to the Floyen until 7 o'clock is every 10 to 20 minutes. So you should time your departure, because we will start the aperitif at 7 o'clock. And also another thing is that the funicular has a line. So there's a line to the ticket, and also you can just scan your QR, so you can skip this line to the ticket booth because you already have your own ticket. And after 7 o'clock, coming down from the mountain, the funicular will be every half an hour. So at the top of the hour and the half of the hour. So we should keep in mind the departure time. So if you have something you need to do. And so with that, I hope to see you at the restaurant. There's also a playground up there, so if you are early, you can join, take some photos and have some fun. Okay, see you.